My name's Sarah Moody. So passing of intact genetic information to our offspring is essential for health and fertility. Every cell within us is identical and with information passed on from mother and father. But it is epigenetics, the set of instructions sitting above our DNA, switching genes on and off, that contributes to the fate of the cell. Our sex cells are particularly important as these will mature into sperm and eggs, hopefully creating healthy new life. I'm particularly interested in how the genome in male sex cells is protected during fetal life to ensure health and fertility. During development, DNA is extremely vulnerable as it undergoes a period called the rewriting of the genome. This is when specific epigenetic information that is inherited from both our parents removed, and then they're re-established in a manner specific to the sex of the baby. So during this time, the DNA is open and exposed, and it's vulnerable because there are elements within our DNA called mobile elements that can move around and potentially cause mutations leading to cancer, disease, or infertility. The pathway I'm interested in is the pRNA pathway, and it's involved in the repression of these mobile elements. For example, these gardeners, or the components of my pathway, are planting trees to protect the area. In a cell, these trees would be methyl groups added to DNA to prevent access to genes. And by doing this, represses the mobile elements, leading to healthy, happy offspring. However, if this pathway isn't working, we get a loss of methylation, or a loss of vegetation, so to speak. And these mobile elements have a clear runway they can take off, move around the genome, potentially causing mutations, leading to infertility and disease. But how does this pathway work? My project is really interested in finding out what motivates these gardeners to protect the male genome during development. One such candidate is activin. It's important for testis development and the establishment of the sex cell population. During pregnancy, women with high blood pressure or preeclampsia are exposed to high levels of activin, so it's vital for us to understand how changes in activin could affect testis development within the womb. I now have evidence to suggest that altered activin signaling can change the components of the pRNA pathway, and my project aims to identify how these pathways interact and work together to protect our future generations. Thank you.